Good day, everyone. So our topic for this module number 8 is about cellular respiration. So as we all know that nutrients and other molecules are being imported into the cell. So inside the cell, they are being metabolized or broken down. And there is a possibility that these are synthesized into new molecules and if needed, are being modified. They are also transported around the cell and possibly distributed to the entire organism. So, for instance, the large proteins that make up muscles are built from smaller molecules imported from dietary amino acids. Meanwhile, complex carbohydrates are broken down into simple sugars that the cell uses for energy. So, in this module, our main topic or main agenda is how the cell convert glucose molecule into energy and how energy, just like ATP, is utilized inside the body. So for our learning outcomes, we have the following. First, explain how ATP is used by the cell as an energy source. Second, describe the process by which energy stored in a glucose molecule can be used by a cell. Third, diagram the major events in cellular respiration as to raw materials used, enzymes involved, end products, and the productions of ATP. Fourth, discuss the fundamental difference between anaerobic cellular respiration and fermentation. And five, describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So let us now begin with the first lesson on this module, which is the ATP. So ATP stands for adenosine triphosphate. So even exergonic, when we speak of exergonic, we are talking about energy releasing reactions require a small amount of activation energy to proceed. However, consider endergonic reactions. In endergonic reactions, which is the opposite of exergonic, endergonic reaction require much more energy input because their products have more free energy than their reactants. So within the cell, where does energy to power such reactions come from? So the answer lies with the energy supplying molecule called as adenosine triphosphate or otherwise known as ATP. So this ATP is very small, relatively simple molecule, but within its bond contains the potential for a quick burst of energy that can be harnessed to perform cellular work. You have to remember that by definition, ATP is the usable energy of the cell. So when we speak of usable energy of the cell, that means that it serves as a currency of energy within the cell. So thus that the different activities of the cell will only be powered by this adenosine triphosphate or ATP. So this molecule can be thought of as the primary energy currency of cells, as I mentioned a while ago, in the same way that money is the currency that people exchange for things they need. So ATP is used to power the majority of energy requiring cellular reactions. So since we are talking about biochemistry, we are going to describe and explain the chemical aspect of ATP as well as that of the cellular respiration. So let us talk about ATP in living systems. So a living cell cannot store significant amounts of free energy. Excess free energy would result in an increase of heat in the cell, which would denature enzymes and other proteins and thus destroy the cell. So that's one thing that we can consider, no, that ATP cannot just be stored, because since ATP is, or the energy, no, will make the cell hot, and that a particular situation, it will damage the enzymes in the cell as well as the other proteins, and then the cell will be destroyed. So, rather, a cell must be able to store energy safely and release it for use only as needed. So, living cells accomplish this using ATP. The one that I explained a while ago, the excess free energy, is energy in general, but not referring to ATP. Free energy in general would result in an increase of heat in the cell, and that's why we have ATP, because ATP can be stored, and ATP will not lead to heat in the cell. So living cells accomplish this using ATP, which can be used to fill any energy needed by the cell, and that is the question there is how. So it's the same as the function of that a rechargeable battery. So when we use a rechargeable battery, whenever we use it, we just plug it and then it became low bat, and then we recharge it again, and then we can still continue using it. So when ATP is broken down, usually by the removal of its terminal phosphate group, energy is released. So ATP can be converted to ADP, or known as adenosine diphosphate. And then ADP can still be further broken down into AMP, or adenosine monophosphate. So the source of the energy is actually the bond between the phosphate group. So this energy is used to work by the cell, usually by binding off the released phosphate to another molecule, thus activating it. So for example, in the mechanical work of muscle contraction, ATP supplies energy to move the contractile muscle protein. So as you can see here on the illustration, so we have here the chemical composition of the ATP. So at the heart of ATP is a molecule known as the adenosine monophosphate, or AMP, which is composed of an adenine molecule bonded to both a ribose molecule and a single phosphate group. So if you will recall, adenine is one of the nucleotide in the nucleic acid, and ribose is a sugar. Ribose is found in RNA. And then you have there the alpha, beta, and gamma phosphate group. So phosphate group is composed of phosphorus and oxygen. So these bond with one another, like for example, alpha and beta and gamma. So these bonds are, when broken down, can be able to release energy, and that's the ATP. And that's the energy needed by the cell. So the addition of a second phosphate group to this core molecule results in adenosine diphosphate, and the addition of third phosphate group forms adenosine triphosphate. So what is the role of ATP in bioenergetics? So according to Lippmann, no, so Lippmann introduced the symbol P, indicating high energy phosphate bond, and the term group transfer potential is preferred to high energy bond. So that's why among the three forms of adenosine phosphate, the one with the highest amount of energy is adenosine triphosphate, or ATP, because there are two bonds between phosphate group, and these bonds are the ones releasing energy. That's why the term is high energy bond. So the phosphate bond in AMP is of the low energy type, since it is a normal ester link. And ATP is the donor of high energy phosphate, and ADP can accept high energy phosphate to form ATP. So ATP-ADP cycle connects these processes, which generate P to those processes that utilize phosphate. As you can see on the illustration, we have there the number of phosphate per adenosine phosphate. So adenosine monophosphate containing one phosphate group, adenosine diphosphate, two phosphate groups, and adenosine triphosphate, three phosphate groups. So this time, let us talk about the coupling reactions. So in this topic, we have this endergonic and exergonic processes or reactions. So endergonic and exergonic are two types of chemical reactions or processes in bioenergetic reactions. So the names describe what happens to energy during the reaction. And the classifications are related to endothermic and exothermic reactions, except that endergonic and exergonic describe what happens with any form of energy, while endothermic and exothermic relate only to heat or thermal energy. If you are going to recall, no, endothermic and exothermic topics are found in the senior high school sciences and sometimes also in the junior high school sciences. So endothermic and exothermic can be associated with heat transfer and or thermal energy transfer. Okay, so let's talk first about endergonic reactions. So as mentioned, endergonic reactions may also be called an unfavorable reaction or non-spontaneous reaction. So when we use speak of non-spontaneous, so it is not spontaneous, it is not continuous. So for it to happen, there is an energy requirement. Unlike if it is spontaneous, wherein there's like that domino effect. But if it is non-spontaneous, so for it to happen, for the reaction to happen, there should be energy. There should be a requirement. There should be energy to it. So endergonic reactions absorb energy from their surroundings. So the chemical bonds that are formed from the reaction are weaker than the chemical bonds that were broken. So in here also, we are going to discuss some about Gibbs free energy and entropy. So the free energy of the system increases. The change in the standard Gibbs free energy, or capital letter G, of an endergonic reaction is positive or greater than zero. And the change in entropy S is decreasing. So that's why, as mentioned, endergonic reactions are not spontaneous. So examples of endergonic reactions include endothermic reactions, such as photosynthesis. In photosynthesis, it is endothermic because you'll be requiring energy from the sun for it to happen, as well as melting of ice into liquid water. So that's why ice melts, because it absorbs energy from the surroundings. So if the temperature of the surrounding decreases, the reaction is endothermic. So in endergonic reactions, as shown in the illustration, it requires energy and usually in the form of ATP. So the opposite of endergonic reaction is known as exergonic reaction. So it may be called a spontaneous reaction or a favorable reaction. So exergonic reactions release energy to the surrounding. So meaning to say that it is the opposite of that of the endergonic reaction. So the chemical bonds formed from the reaction are stronger than those that were broken in the reactants. So the free energy of the system decreases. The change in the standard Gibbs free energy, or capital letter G, of an exergonic reaction is negative or less than zero. So the change in entropy S increases. Another way to look at it is that the disorder or randomness of the system increases. In exergonic reaction also, it occurs spontaneously, so there's no outside energy required. Examples of exergonic reactions include exothermic reactions, such as mixing sodium and chlorine to make table salt, combustion, and chemiluminescence, wherein light is the energy that is released. And if the temperature of the surrounding increases, the reaction is exothermic. So as you can see on the illustration of the exergonic reactions. Okay, so this time let's talk about the third lesson, which is cellular respiration. So as mentioned, cellular respiration is the process by which living cells break down glucose molecules and release energy. So cellular respiration is far different from the usual respiration that we know, wherein by definition, when we speak of respiration, it is the exchange of gases between your lungs and the atmosphere. We give off carbon dioxide and then we absorb oxygen. On the other hand, this cellular respiration actually is a continuation of that, wherein we use oxygen so that we can be able to convert um, our glucose molecules into a usable energy. So, meaning to say that um, the process is similar to burning, although it doesn't produce light or intense heat as a campfire does. So, this is because cellular respiration releases the energy in glucose slowly in many small steps. So, that's why in the conversion of glucose into ATP, there are variety of steps, actually complicated steps or small steps, so that the cell will not be bombarded with lots of ATP. So meaning to say that glucose molecule has to undergo these processes because there's a need to, and it's for the welfare of the cell itself. So if the cell will be properly nourished, then the entire organism will get benefit from it. So it uses the energy that is released to form molecules of ATP, the energy carrying molecules that cells use to power biochemical processes. So that's why even though a person, let's say a dead organism, intake food for some reason, let's say you injected, let's say glucose molecule or any kind of food to a dead organism, so the food will be useless simply because the cell would require not only food but also other things for the cellular respiration to occur. So by summary, we can use this chemical equation, which is C6H12O6, or the glucose molecule plus 6 atoms of oxygen can form 6 atoms of carbon dioxide, 6 atoms of 6 molecules of water, and the energy or the ATP. As you can see here, that what is essential in cellular respiration, especially if it is aerobic respiration, is the presence of oxygen. So if there's no oxygen left, for example, so this will not um, be implemented, or it will not happen. So cellular respiration has 3 stages. We have here glycolysis, the Krebs cycle, also known as the citric acid cycle and the ETC, or the electron transport chain. Or sometimes it is better to call it as oxidative phosphorylation. So cellular respiration occurs in the cells of all living things, both autotrophs or plants and heterotrophs, animals, consumers. All of them burn glucose to form ATP. So this is the aerobic respiration, a kind of cellular respiration. No, the opposite of this is anaerobic respiration. So we can just simply deduce the explanation by saying that aerobic respiration is the conversion of glucose molecule into energy with the presence of oxygen. On the other hand, if it is anaerobic respiration, um, it is in the absence of oxygen. So as you can see here on the illustration, that the stages of the aerobic respiration happens in the different parts of the cell. For example, glycolysis, the basic stage, happens outside the mitochondria. And then citric acid cycle or Krebs cycle and oxidative phosphorylation happen inside the mitochondria. Glycolysis happens in the cytosol of the cell. So as you can see here also, that there are common products in each stages or in each stage of the aerobic respiration. So let us now begin with glycolysis. So as you can see here on the illustration, that the main purpose why there is a need for glycolysis is that the glucose molecule has to be converted into pyruvate or pyruvic acid. So the glucose molecule here is bombarded with phosphate group. So the phosphate group came from that of the adenosine triphosphate. So the phosphate group is then attach itself to the sixth carbon of glucose. So that's why the term is glucose 6 phosphate. So same thing with fructose 6 phosphate. So from glucose 6 phosphate, it is converted to fructose 6 phosphate because of the instability of the glucose 6 phosphate molecule. And then there's another bombardment of ATP, getting one phosphate group. So that's why you call it now as fructose 1,6 biphosphate. Biphosphate or diphosphate is the same. Then after that is the utilization of the HAP and G3P, wherein I'll be explaining that later on. And then there is another bombardment of phosphate group. That's why the term now is 1,3-biphosphoglycerate. And then this 1,3-biphosphoglycerate is then converted to 3-phosphoglycerate, and then 2-phosphoglycerate, and then phosphoenol pyruvate, leading to pyruvate or pyruvic acid. So on the right side of the illustration is the different enzymes that are needed in each stage of the steps of glycolysis. So let us look into the chemistry aspect of the glycolysis process. Look at this one. So this chemical reactions is they are the same as that of the previous slide. It's just that in here we can be able to see the different structures of the glucose molecule as well as the different molecules as they are converted from one form into another. For you to better understand this, you have to take into consideration reactants and products. So on the first illustration, on the first part, this one, this is the raw material glucose, which is the first part of the glycolysis. So glucose molecule came from the food that we eat. This is the smallest or the monosaccharide sugar. And then there is the bombardment of the ATP. So ATP is now then converted to ADP as the the energy do that, or the ATP do that, so it releases energy. So the phosphate group is then attached itself on the sixth carbon. So that's why the term is glucose 6 phosphate. And then on the second step is that this entire glucose 6 phosphate now is known as the reactant, so that the product will become fructose 6 phosphate. So there is a difference in the structure, but it's the same thing, no? Though the name was changed into fructose from glucose. And then there's another bombardment of ATP here. So that's why there are now two phosphate group attached to it. So we have here one, and then we have another one here. So we call it now as fructose 1,6 because this one is attached on the first carbon and the other one is attached on the sixth carbon. So biphosphate or diphosphate, they are the same. And then upon doing that, no, the fructose 1,6 biphosphate will now be broken down into two equal parts. So the first part is will now be called as glyceraldehyde 3-phosphate, and the other one will now be called as dihydroxyacetone phosphate. So we are going to move on the left side of the illustration. You have here now the product, the reactant. So this is the reactant, and then this is the product. Okay, so for example, we have here glyceraldehyde 3-phosphate. If you are going to recall this in the previous topic, glyceraldehyde is a sugar, no, with an aldehyde chemical formula. So it is now then converted into 1,3-biphosphoglycerate. And then this one will now become the reactant, and then this will now become the product, 3-phosphoglycerate. And then another conversion is the 2-phosphoglycerate, and then phosphoenol pyruvate, and then pyruvate, right? So what we can highlight here is the utilization of ATP. So for glycolysis to happen, there is a need to invest ATP to gain more ATP. So we can also see how ATP, or the phosphate group from ATP, is used. What's the purpose of it in the molecule, and what's the effect of it? For example, the attachment of glucose, of phosphate in the 6 carbon of glucose in the equation 1 lead to the formation of glucose 6 phosphate, and glucose 6 phosphate lead to the formation of fructose 6 phosphate. And then at the end of it, pyruvate was formed. So you have to remember that in this equation, there will be two pyruvate or pyruvic acid, since there are two um, 3-carbon molecule formed from fructose 1,6 biphosphate. So the first one is glyceraldehyde 3-phosphate and the other one is dihydroxyacetone phosphate. Thus, the cell utilized 2 ATP, but the cell produce 4 ATP. So from there, we can say that there is an earning of 2 ATP after each glycolysis reaction. So the next one is the Krebs cycle. So Krebs cycle now happens inside the mitochondria of the cell, wherein pyruvate or each molecule of pyruvate is then converted into acetyl-CoA or acetyl coenzyme A. So this acetyl coenzyme A will now be used as a material in the oxidative phosphorylation, the last stage in the cellular respiration. So you have also to be familiar with the different acronyms like NAD, nicotinamide adenine dinucleotide. There is plus because it is charged. And then you have also here nicotinamide adenine dinucleotide or NADH hydrogen. Okay, so let us look into the chemical aspect of it. So the first one is the condensation of the pyruvate into citrate. So there's condensation, and then you have here dehydration, and then hydration, oxidative decarboxylation, oxidative decarboxylation again, then substrate level phosphorylation, dehydrogenation, hydration, dehydrogenation. That's why it is a cycle or citric acid cycle, because it is a continuous loop of reaction. So from condensation, acetyl-CoA is then converted into citrate. Okay, so the citrate will undergo dehydration or the removal of water. So since there's a removal of water, citrate became aconitase. And then aconitase will undergo hydration or the addition of water, and then it will now become isocitrate dehydrogenase. This one. And then you also have here oxidative decarboxylation. Decarboxylation is known as removal of carbon dioxide. So there is this arrow, removal of CO2. And then the chemical structure became like this. It is now known as alpha-ketoglutarate. And then from here, from isocitrate dehydrogenase, you can be able to utilize NADH, so that you can easily go to the step 8. And then you also have substrate level phosphorylation, and then dehydrogenation, and then hydration, and then another dehydrogenation. So that makes the Krebs cycle. All right. So let us now move on to the last stage, which is the oxidative phosphorylation. For oxidative phosphorylation, it is done in order to couple the redox and phosphorylation reactions needed for ATP synthesis in the body. So in cells, this is accomplished through an elegant proton pumping system. So this elegant proton pumping system is available in our cell, particularly the mitochondria. So a number of proteins are required to maintain this proton pumping system and catalyze the oxidative and phosphorylation reaction. So as you can see, the oxidative phosphorylation are divided into electron transport chain and then the chemiosmosis. So what uh, the products in the Krebs cycle, like that of NADH, which is an energy carrier of the cell, and then FADH2 are now needed in order to pump protons. So electrons are transferred from NADH through a series of electron carriers to oxygen. So the electron carriers are proteins embedded in the inner mitochondrial membrane. So there is also the transfer of electrons by these carriers generate a proton gradient across the inner mitochondrial membrane. So in short, there's a need for the oxidative phosphorylation because oxidative phosphorylation is the last stage of cellular respiration, and that means also that um, that the highest number of ATP is yield, no, in this last stage. So as mentioned here, there are 30 to 34 ATP. So in lesson 4, we have here anaerobic respiration in the name of lactic acid fermentation if it is animals, and then we also have alcoholic fermentation for plants. So the ethanol, the component of our rubbing alcohol, actually is a product of alcoholic fermentation, right? So that ends my discussion. So see you again by next time.